Hello, students. Have a great day. Today, we will discuss the new topic mitosis. The previous section, we learned about the cell cycle different phases, and today we will discuss about mitosis different phases. As we know that mitosis consists of two substages. One is karyokinesis, and the second is the cytokinesis. Always remember, karyokinesis is followed by the cytokinesis. What is karyokinesis? It is the division of the nucleus and followed by the division of cytoplasm, which is known as cytokinesis. Okay, so we will first discuss about the different points under the karyokinesis. Karyokinesis consists of again four phases. One is the prophase, then metaphase, third is the anaphase, and the fourth one is the telophase. Let's start with the first phase, that is the prophase. Prophase, there are certain key events taking place in the prophase. This is the very first phase in the mitosis. In this, chromatin condensation taking place. Okay, what is chromatin? You know the difference between the chromatin and the chromosomes. Chromatin condensation. Chromatin is a thread-like structure. Okay, and the interface, the two chromatin threads are there, and these two chromatin threads this undergo condensation to form the chromosomes. Okay, so the chromosomes are formed here in the prophase. So thickening of the chromatin fibers, the chromosomes taking place in the prophase stage, and in the before the cell division in the cell surrounded by the nucleus, right? And this nuclear envelope, they will disintegrate during this prophase. Okay, so the disintegration of nuclear envelope, this is a nuclear envelope that will disintegrate. Envelope disintegrate the prophase. And asters there, asters or the astral fibers with the spindles there. Okay, spindle fibers move towards the poles. They will move towards the polar region cell. So these three key events taking place during the prophase stage. So prophase, remember, condensation of the chromosomes, nuclear envelope disintegrate, and the spindle apparatus migrate towards the poles. So remember all the three events. Moving on to the next phase, that is the metaphase. The second phase is the metaphase. Just to have a look at the diagram here, you can see that is chromosomes. Okay, so these are the chromosomes there, and centrioles. From the centrioles, spindle fibers arising. Spindle fibers. So very first, the chromosomes, all the chromosomes in the cell, they arranged at the equator of the cell that form a specific region, and this is called a metaphase plate or equatorial plate. Metaphase plate or equatorial plate. Okay, this is a 1 mark question. What is metaphase plate or equatorial plate? During the metaphase, all the chromosomes are aligned at the equator of the cell, and that form the metaphase plate. Equatorial region called a metaphase. What is the purpose of alignment to at the equator? For the equal distribution of the chromosome daughters. And then just check there here the spindle fibers, these spindle fibers moving towards the chromosomes, and we know that the chromosomes, they are attached at the center. That centromere is there. Next to the centromere, there is a protein structure that is called a kinetochore. Kinetochore is there. So these spindle fibers attached towards the kinetochore. Kinetochore is placed here. Here only the spindle fibers will be attached. So kinetochore protein at the centromere captures the spindle fibers from the opposite pole, creating the tension that maintains this alignment. I hope this part is clear. So metaphase, remember the key events. Chromosomes achieve perfect alignment at the center with the spindle fibers creating a balanced tension. Okay, then moving on to the third phase, that is the anaphase. Anaphase. During the anaphase stage, very first there is the centromere splits, splitting of centromere and separation of the chromatids taking place. So separation of sister chromatids taking place. That means the chromosomes to chromatids are there. They are joined at the centromere, and these are the sister chromat. So anaphase stage, what happens? This centromere splits and two sister chromatids separated. And then the next stage is these sister chromatids move towards the poles. And this movement of the chromatids towards the pole are due to the shortening of the spindle fiber, so that it is pulling the chromatid towards the cell poles. Okay. Then finally what happens? There is the all these chromatids, the spindle poles will move up towards the polar region, and there is the chromosome will be set the polar region. Okay, so just have a look at the diagram here. Compare the previous diagram. In the previous diagram, at the equator, all the chromosomes aligned. Now in the equator region, it is empty, right? The metaphase plate is not there. You can see the separation of the chromatids, and where it is going, these chromatids are going towards the pole, and that movement is due to the shortening of the spindle fibers. Remember these key events during the anaphase. The chromatids move towards the opposite pole, and also the spindle fibers shortening. And one more point is there, the elongation of the cell taking. Okay. The cell elongates little more during the anaphase stage. And the last phase under the karyokinesis, the fourth phase, is the telophase. So telophase stage, whatever we learned in the prophase, when you compare, exact opposite taking place here. So chromosomes here, decondensation taking place. The decondensation of the chromosome to form the chromat. Here, the thickened chromatids, they undergo decondensation to the chromatin threads. Okay, so in the prophase, there was a condensation from the chromatin to the chromosome. Here in telophase, decondensation of the chromosome to chromatin. And in the prophase, there was a disappearance or disintegration of the nuclear envelope. Here it is the reformation of the nuclear envelope. The nuclear membrane assembled to cover the chromosomes, right? Then the spindle fibers, now there is no use of spindle fibers because already the nucleus and all nuclear material separated. So spindle fibers slowly disappear, break down, and disappear during telophase. Okay, so these are the four phases under the karyokinesis. Remember, mitosis, karyokinesis, cytokinesis there. Karyokinesis again divided into four phases: prophase, metaphase, anaphase, and telophase. I hope this part is clear for all of you. Moving on to the last part of the mitosis, that is cytokinesis. That is a final separation. What is cytokinesis? Cyto is related to cytoplasm. Okay, so the division of the cytoplasm, that is called a cytokinesis. And when you compare the animal cell and plant cell, there is some difference in the cytokinesis, right? So just have a look at these two diagrams. In animal cell, first there will be cleavage furrow formation taking place from the cell membrane part. Okay, so in the animal cell, there is a cleavage furrow formation taking place from the cell membrane. From the cell membrane. At the same time, at the center here, small contractile rings will be formed. And this cleavage furrow slowly moves towards the center from both the sides and then completed, and then the two daughter cells are formed. The cells are formed after mitosis. Already there were karyokinesis over and two nuclei formed, but still it's a single cell only. And the cytokinesis is cytoplasm divides as a result. There are two daughter cells, complete separation taking place. Coming to the plant cell here, instead of cleavage furrow, there are cell plate formation taking place. Cell plate formation. What is cell plate? There is a structure formed at the equator, the center of the plant cell. It is actually formed from the small vesicles called phragmoplast. Phragmoplast. And this phragmoplast is actually formed from the Golgi complex. From the Golgi only, small vesicle formation taking place, and that they are called phragmoplast. This phragmoplast join to form these vesicles only called a phragmoplast. When they join to form a single structure, then it is called a cell plate. Cell plate formation at the center, and then it grows towards the periphery. Outward growth will be there, and finally it will join with the cell wall, and there is a separation of the cytoplasm, so two daughter cells are formed. So remember the cytokinesis in plant cell and animal cell. Okay. Distinguish between the cytokinesis in plant cell and animal cell. So in animal cell, contractile ring of actin myosin filaments are formed, and then there is a cleavage furrow formed at the cell membrane that grow inward until separation occurs. Then plant cells, there is a cell plate formation taking place from the small vesicles. And what is the name of the vesicle? It is called a phragmoplast, that is formed from Golgi complex. And these cell plates growing outward until it fuses with the cell wall to create the two separate cells. Okay, so that's all about the mitosis. Mitosis consists of karyokinesis and the cytokinesis. I hope this topic is clear for all of you. We will meet in the next video. Till then, thank you. Bye.